Thank you, Brenda. And I would like to say thank you to the SBIA group for this opportunity for DMA to have a webinar and to continue our outreach efforts in terms of sharing information that we have here in our Division of Microbiology Assessment. Brenda also asked me to say a few words about my own background before I get going. And so for 14 years, I was a college teacher, teaching microbiology and related courses in both upstate New York and Massachusetts. And I came to CDER to become a review microbiologist in 2003, which has been a wonderful experience for me. I've really am thrilled to work for an agency with such a wonderful public health mission. I'm very excited to present this case study to you today because we in the Division of Microbiology Assessment feel that this is a very important case study to get out information and to illustrate why FDA has such concerns regarding Burkholderia Cepacia Complex. Now you can see from the title this refers to Burkholderia multivorans. And just as a point of information, when Erica was presenting regarding Burkholderia Cepacia Complex, that term complex refers to multi-species that are all very related to one another. Burkholderia multivorans is one of the species that belongs to Burkholderia Cepacia Complex. So Erica's presentation really provides great background for this one because you'll notice from her presentation that we at FDA have had recent significant health-related issues regarding this particular organism and a number of different types of products. Okay, so my first slide is an FDA disclaimer slide. And then what I want you to focus as we work our way through this case study is the following. These are the themes that will become very important. First is the relationship between BCC and pharmaceutical water systems. Again, Erica laid the foundation for that in her presentation. The idea that BCC can form biofilms. So the organism is capable of formation of biofilms in the plumbing of your water systems. The notion that BCC is capable of developing resistance to antimicrobial preservatives. Again, Erica mentioned this, and related, BCC is able to develop resistance to antimicrobial drugs as well, which becomes problematic once the organism is in patients. And finally, all of these things make the organism a concern regarding aqueous non-sterile drugs. I won't belabor that point any further because Erica spoke quite a bit about water activity and why we have concerns at FDA regarding the aqueous non-sterile drugs. So I do want to begin with two Code of Federal Regulations citations that have to do with this case study. The first is 211.113, control of microbiological contamination. The CFR here states that appropriate written procedures designed to prevent objectionable microorganisms in drugs not required to be sterile shall be established and followed. Okay, so what this citation says is that if you're going to make a non-sterile drug, you need to have written procedures to keep objectionable organisms out of it. Second, 211.165 regarding testing and release for distribution. This says there shall be appropriate laboratory testing as necessary of each batch of drug product required to be free of objectionable microorganisms. So what this says is that you should have a test at release that demonstrates that your non-sterile drug does not have objectionable microorganisms in it. Which leads me to my polling question. So I wanted to get a little bit of feedback from you. And the question has to do with, if you're a pharmaceutical microbiologist and you work on both the non-sterile drugs and the sterile drugs, do you consider your non-sterile drug work to be more challenging than your sterile drug work? My interest in asking this question has to do with the fact that we consider the sterile drugs, of course, to be the most risky to patients because of the way that those drugs are administered. And I'm seeing it looks like, in terms of whether you agree or disagree, it looks like more of you agree than disagree, and many of you are not a microbiologist. So let's go back to the presentation. Again, my reason for asking that question is that even though the sterile drugs are the drugs that we focus on the most in terms of microbiological quality, those of you that work in the industry on pharmaceutical products, you do struggle with this non-sterile area. And the reason is that the non-steriles can be released with a measurable bioburden, but it can't have too much, and it can't have the wrong ones. So that, I know, presents challenges to you. Okay, so I've got a slide here that references Section 314.81 of the CFR. This is the section that describes field alerts. I included this because this case study at FDA began with a field alert report that was sent into the agency. So I wanted to provide a little bit of background about what a field alert report is. When you go through your slide package, you'll see that what this points out is that if your product contains bacteriological contamination or some other significant issue, such as a chemical or physical problem, you need to report this to the FDA using a field alert report. And I won't belabor these points. I give you the information so that you have them in your slide package. You can find a field alert report at the following website. On the FDA website, it's FAR Form 3331. And I did want to include a slide that shows what a field alert report looks like. And my point for sharing this with you is that once we receive these at FDA, we don't have a lot of detail to go on. So we find out about what the drug is, which lots have a problem. We find out what you've identified as the problem. There may be a root cause in there, but the reality is that not a lot of information comes in other than to alert us to the fact that there's a problem with the batch. So for us in the Division of Microbiology Assessment, the case study began with a field alert report that was sent to us by OPQ's Office of Surveillance. So our colleagues in the Office of Surveillance contacted the Division of Microbiology Assessment, and they said, we have a field alert report. It's attached to this email. It told us that FDA is meeting with a firm to discuss their investigation and plan relative to this field alert report. The Officer of Surveillance asked DMA, could you provide some subject matter expertise in the way of questions for discussion with the firm? At the same time as Officer of Surveillance was reaching out to DMA, they also reached out to the Clinical Review Division where this product is reviewed. And I like to emphasize this in that the Officer of Surveillance wanted an opinion specifically from the clinicians here in CDER who review the drug. And the question for them was, does the presence of this organism, Burkholderia multivorans, in the subject drug product present a risk to patients? So my point for sharing that with you is that that question doesn't get directed to the quality microbiologists, which is the group that Erica and I are part of. I certainly have my own opinion on that, but the important point here is that CDER goes right to the medical officers in this case and says, does this organism present a patient risk? And the clinical review division says, yes, in this case, this is a risk to patients. So what did we learn when we read the field alert report? We learned, first of all, that this was a nasal spray product. It was approved in the 1990s. Okay, relatively speaking, that's an old product. When I say relatively speaking, I'm meaning in my experience. We learned that it was an aqueous product, and it's preserved with benzalkonium chloride. Okay, so Erica spoke along quite a bit about what aqueous products are in terms of a microbiological risk. This one contains benzalkonium chloride as a preservative. We also learned that the firm, the company, determined they had 2 batches that were positive for Burkholderia multivorans. The FAR indicated that these 2 batches were still in their control. Okay, that's good news to us. And the FAR also indicated that they had performed expanded testing on 10 batches. Okay, and I put expanded testing here in quotes because the nature of how they did that testing was not made known to us in the field alert report, but they had tested 10 batches, and very concerning results to us. Notice that 5 that had been previously tested negative were now testing positive. Red flag goes up in our minds. That's a concern. So the firm, again, telling us that here's 10 batches that initially had been negative, and now 5 of those were positive. Very concerning. So at this point, I want to give a shout out to my close colleague and branch chief, Steve Langell. Steve and I worked together on this case study. We put our heads together and came up with questions as requested by our colleagues in Office of Surveillance. Here are the questions. And I do want to say, I know that these slides are wordy. That's the reason for that is that I want you and the audience to take home a direct copy of what went back and forth between FDA and this company. So there's a lot of words here, but again, that's for you to digest after our webinar today. First question, how were the initial batches of the drug product determined to contain Burkholderia multivorans? Was this demonstrated following testing of the drug product according to USP 61 for total aerobes or using a Burkholderia specific test? What is the concentration of Burkholderia per ml of the drug in these batches? Next question. Regarding the additional 10 product batches that underwent expanded testing, how is the expanded test different from the test performed at release? Again, we didn't know anything about the expanded test, so we asked the question. Additional questions. Is the water system that's used to manufacture this drug routinely tested for organisms that belong to Burkholderia Cepacia Complex? We recognize that the investigation of this incident has not yet determined a root cause. Summarize the steps of the drug product manufacturing process that you have tested for evidence of this organism. And then finally, what is your plan for the drug product batches that contain Burkholderia multivorans? This last question, it wasn't made known to us in the field alert report what the company had planned in terms of disposition of the 2 batches that were positive, and so we felt a need to ask this question. So these questions are compiled. They're sent to the company prior to the teleconference, so the company has some time to digest, gives us an opportunity to have a fruitful teleconference. And then let's discuss the bullet points in terms of what we learned during that teleconference. So this is the TCON between the company and FDA. We learned that Burkholderia was picked up using a bile-tolerant gram-negative test performed according to USP 62. And I'll also share with you, this is what we learned during the TCON. As we dug into this case further over the next week, we also found out that the company was able to pick up this organism using the Pseudomonas aeruginosa specific test in USP 62. We learned that the batches were too numerous to count. Wow, that was a surprising result. That's a lot. We learned that the firm had already performed an investigation. And what they had found in their investigation was that their purified water system had developed a biofilm. And the firm explained to us that this particular part of the purified water system, there was a small portion of it which was poorly engineered, meaning the water wasn't draining well from that particular part, and it was undergoing an improper sanitization process, and Burkholderia formed a biofilm. This was a point that Erica brought up in her presentation. This is a point that is well known. These organisms are capable of forming biofilms in the piping systems. The firm also told us during this teleconference that their system was in control at the time of the U.S. manufacture of the U.S. batches. So immediately after the teleconference, FDA had an internal meeting. And you see here a list of folks from FDA that were part of that meeting. We had CDER Office of Compliance, we had Office of Surveillance, we had the district office from ORA, the district office where that manufacturing site is located. And then finally, we had the Division of Microbiology Assessment at this meeting. The question on the table for us was, do we need to recall the 58 batches that are here in U.S. commerce? So to summarize the case study to this point, we knew that the company had a problem with a contamination of several batches. The firm told us that all of those batches were still in their control, but we also knew that there were 58 batches which were here in the U.S. And the question for us was, do we need to recall? Here are my comments from that meeting. Well, my first comment is the product was approved a while ago, back in the late 1990s. The reason that that's significant is that we had no record of an FDA microbiology review. Why is that important? Well, first of all, just let me explain that in the 1990s, and certainly today it's true as well, most of our effort in DMA is focused on the sterile drugs because those do present the highest risk to patients. At this time, we do have the resources to review non-sterile aqueous products, and we do that now in 2017. However, back in the late 1990s, microbiology review of these products was not being performed because the resources allowed for review of sterile products only. So without that micro review, I had nowhere to go in terms of trying to find out how the product is made regarding the microbiological in-process controls and testing. These are all the things that were therefore unknown to me. Are all the batches subject to micro-testing at release? I knew that the company was doing some testing because they came to us with this information. However, I didn't know whether they were doing skip lot testing. And if they were testing everything, what method were they using? Was it consistent with the compendial methods? Finally, since the product is preserved, the question became, are the test methods suitable for use with the subject drug product? So this is method suitability. The idea of method suitability demonstration is very important when performing microbiological tests. This is the idea that you can get out organisms from a preserved product or any drug product if they are present. Didn't know any of this. As a result, we had more questions for the firm. And so these questions were directed at trying to make an assessment as to whether a recall was warranted. Regarding the 58 lots of the drug currently in the U.S. market, provide us the test methods, the acceptance criteria, data summaries from all micro-testing performed on the drug product at release. Include data summaries demonstrating that the micro-test methods are suitable for use with the drug. Provide the stability protocol. Provide data summaries from micro-testing to date on product in the U.S. market. Now, from those questions, what came back to us were literally hundreds of pages of reports from this company. And so what I'm going to share with you now is the bullet points that came out of our review memo that's archived in CDER. So these are direct quotes from our CDER memo after review of hundreds of pages of reports from those questions. First of all, and I can summarize quickly by saying the firm had done everything right both before they had the problem and afterwards in terms of a testing regimen for this product. Bullet 1, the firm routinely performs micro-testing on this drug in excess of what is recommended in USP 1111. Erica had a slide on USP 1111. In this case, when you see that I'm stating here that they test in excess of what's recommended there, I'll tell you that this company was testing for 5 specified organisms out of USP 62, whereas USP 1111 suggests only testing for 2. So they were going above and beyond. The micro-release testing performed on the drug is performed according to 61 and 62. So the firm does follow the compendial testing methodology. The firm has satisfactorily performed testing to demonstrate that the methods are suitable for use. Again, that's a huge piece to this, knowing that the test method is valid. They had also performed testing to demonstrate that they could get Burkholderia multivorans, the contaminant, out of their test methods. The micro-release testing on these 58 batches in the U.S. market met acceptance criteria and was acceptable. Micro-testing of samples in the stability program is routinely performed. The stability data meet acceptance criteria and are acceptable. Okay, these are, again, all bullets coming out of our CDER review memo. My final summary to CDER starts off with a disclaimer. And yes, even communicating to other people at FDA, I'll use a disclaimer. This reviewer acknowledges that end product release testing presents limitations with regard to predicting quality of a given batch. However, the information provided to the agency by the firm regarding the micro-release and stability testing does not suggest that a product recall of these batches is warranted from the standpoint of microbiological contamination. I also wanted to share several interesting findings that the firm uncovered during their investigation. They performed a study to evaluate the growth potential of this contaminating bacterium in the drug. Of note, When they spike the organism into the drug, you see that the counts decrease over the first few days. On day 3, the organism begins to grow in log phase. By day 7, counts are up above 10 to the 5 CFUs per ml. Okay, this is shown graphically. This is a graph that came right out of the reports that the firm sent us. And I want to emphasize, FDA did not ask them to do this study. The firm did this study on their own. They took their own initiative to do this. You see during the first 24 hours, those counts decreasing because, remember, the drug product is preserved. However, it doesn't go down to 0, and before you know it, it's in log phase. So after a week, you've got more than 10 to the 5 CFUs per ml in a preserved product. Again, this point that we wanted to emphasize about Burkholderia and its ability to grow through preservatives. What did the firm learn? This provides them with an understanding of the organism in the product. It may explain picking up the organism using the expanded test. I'll elaborate more on that in a moment. It provided the company with an avenue for corrective actions regarding future micro-testing of this product. We'll see that in a moment as well. But I'm emphasizing here, doing this study taught the firm a lot. The second point that I want to make regarding their investigation, the company did testing on numerous retained samples from batches that were here in the U.S. The sequence of the testing was they did testing of 10 batches. 5 of those were positive. We pointed that out earlier. They went back in time and took 25 marketed batches that were manufactured prior to those original 10 and tested those. None of those batches tested positive. That was important information for the firm and for FDA. The information from the expanded testing of these 35 batches was able to point to a time frame when that biofilm formed, and it was able to provide some assurance regarding patient safety and product that was here in the U.S. market. And this is a graphic illustration that shows the testing and the relationship between older product that had been manufactured versus more recent. So let's start at the bottom of this table, all the way on the left. That column refers to date of manufacture of batches. The blue indicated batches that were still in the firm's control. So starting at the bottom and working backwards, you see numerous batches in their control, and then the white indicates batches that were here on the U.S. market. And you see that in the column next to it with the N's, N refers to whether it was released versus Y. Now, the column that has 2 red cells there, that refers to the release testing. Those were the 2 original batches that the firm discovered Burkholderia in. And then the column all the way on the right, it shows the testing of the additional 10 batches, 5 of which were positive. So as you continue up that scale, you see plenty of green space between the batches that were positive and the batches that were in the U.S. market. Again, an excellent visual presentation to give us some assurance of when that biofilm formed and the safety of batches that were here in the U.S. Again, FDA didn't ask the company to do that. This was their own initiative and part of their own investigation. To summarize, there was no recall. And believe it or not, FDA wants to do everything that they can to prevent drug shortages. So this is a win here when we don't have to suggest to a firm that they should be recalling their products. This was good. In addition, the firm implemented several corrective actions following their investigation. They re-engineered the bad plumbing. They improved the sanitization of their purified water system. I'd like to think that their eyes are wide open in terms of BCC. They told us they were going to perform expanded microbiological testing for a year. Okay, so what they mean by their expanded micro testing, they're actually testing more units than what the USP chapters require folks to test. They also modified the start time of their micro-release testing based on what they found in the growth kinetics study. So if you remember, we saw that when you put the organism into the product, at initial time the organisms go down in numbers, only to come back up. The company, because of their kinetics study, decided that they were going to, for a year, modify their start time of when they were going to take samples for micro-testing. To us, that was a very clever, ingenious step to take in terms of this particular product and the experience that they had. Again, they wouldn't have known to do that if they hadn't taken on that study. So my general comments. Industry has been calling for FDA to base our decision-making on science and risk. We know that. We hear you. In terms of drugs, this means, of course, risk to patients. We understand and we agree. However, I will say, and our experience at FDA is varied. Our experience is that some companies come with lots of data, while other companies don't provide us with the data that we need. So that third bullet, in cases where scientific data are not available, then patient risk can't be assessed by CDER, and questions in our minds come up. Additional general comments related to this: to avoid negative business outcomes, such as delays in drug approvals, FDA enforcement actions, or product recalls, industry needs to be ready to provide us with scientific data when it is requested. In my final comments, CDER microbiologists understand that E. coli happens. And, you know, this bullet point is probably better in a live setting than on a radio show like this one, because I can't hear the rippling of laughter that's occurring around the world right now. I'm hoping that you're not confused by why I'm bringing up E. coli in a discussion of Burkholderia. But regardless, my point here is we understand it is difficult to make drugs, whether they're sterile or non-sterile, with adequate microbiological quality. When I say it's difficult, what I'm referring to is humans walk around carrying microbes. The water systems have microbes. Your raw materials have microbes. It is a challenge. We know that. That being said, patient safety depends on drugs with appropriate microbiological quality, whether that's a non-sterile drug or a sterile drug. So for us, our question becomes, how does your company respond when E. coli hits the fan? And what I'm thinking here when I write this question is, those of you that follow 483 observations, you'll find that our investigators oftentimes determine in manufacturing facilities that an investigation should have been started by a firm, or one may have been started and yet it wasn't followed through on. So the question is, how does your firm respond when problems arise? I do want to thank you for your attention. I provide some reference material here in terms of the CFR citations which are relevant to this case study. And thank you very much.